Hello everyone, welcome back after Thanksgiving. I hope you guys enjoyed. So we have a couple of class left before the final semester, final sem. So we in the beginning of some of my beginning lecture talk about about neural induction. Then we talk about patterning. We talk about neural differentiation, neurogenesis, and axon pathfinding. And after that, we are talking about we are going to talk about the synaptogenesis. So it's a sequential process, starts from induction, then you have patterning, then you have differentiation, neurogenesis, axon pathfinding. Now after the axon find its path target, now it makes the connection, the love. So that is called as connection, that is called as synaptogenesis. So we are going to talk about some of these some of these research from different labs, and this is my also active area of research that I'm currently doing from around 1 decade. So we are going to talk about synapse. So what is synapse? Synapse is a functional connection. It can be a functional connection between neuron to neuron. It can be between neuron to muscle, and it can be also between neuron to gland. So these are the functional connections where a synapse communicate with a other cell, like either it will be a neuron or muscle or a gland through a functional connection that we call as synapse. And these are the fundamental unit of information processing. So here, this is a typical structure from Petru D. Camilli lab. He's a big scientist at Yale. So this picture is from his lab. So here, this is the CNS synapse between the neuron to neuron connection. So I'll be talking about soon to neuron to muscle connection, and we'll see some of these important feature in the synapse. So here, this is a presynaptic cell, which is called as presynaptic terminal, presynaptic neuron, containing the mitochondria, and there are certain structure in the presynaptic compartment or presynaptic cell, and these are electron dense structure. And those if you look at the electron microscope, EM electron microscope, they look like dense. So this is called as active zone, where the synaptic, the vesicle containing neurotransmitter fuse in this zone. That's why it is called as active zone, and release its content through the synaptic cleft. So this is called as the dense-like structure is called as active zone. In Drosophila, it is called as BRP, Bruce pilot. In mammal, it is called as CAST or ELKS-like molecules. So we are going to talk about these things. And the postsynapse, which is magenta color or pink color, this is the postsynaptic cell. And this can be another, like here it is a neuron. So now people say that it is a bipartite synapse, but now they have modified the statement. They're calling it a tripartite synapse. There are 3 cells here. You have presynaptic cell, you have postsynaptic cell, and you have another cell, which is glial cells. And there are a lot of evidence which suggests that glia helps in neurotransmission. Glia supports neuron axonal growth. A lot of active area people are working on. So we are not going to talk about glia thing today, but this is something I want to highlight, that this connection between the presynapse and postsynapse is called as synapse. Okay. So if I'll go detail about how it looks like in the NMJ. So that previous slide was talking about the CNS synapse between neuron to neuron, and here between the neuron to muscle. So this cell is a muscle cell, and this is a presynaptic neuron. Okay. So the presynaptic neuron is same thing shown here. The presynaptic neuron is called as a motor neuron terminal. So this is the axon terminal. So you have the cell body, now you have the axon, now the terminals. So this is the presynaptic terminal, the axon terminal, and this is the postsynaptic muscle. So here in the NMJ, the neuromuscular junction, you can see that in the presynaptic cell, you have the mitochondria. You have these structure that you are seeing, the electron dense structure. This is a cartoonic representation. So looks like T-like structure. This is also called as T-bar or active zone. And the vesicle containing neurotransmitter, they fuse in this region and release its content. And the postsynaptic cell, the muscle cell, is kind of not uniform. They are super fold-like structure. This is called a junctional fold. And in this, muscle cells, you have the receptor present in the muscle cell. These are called as postsynaptic receptor. So depending upon types of synapse, it can be like GABA receptor, or it can be glutamate receptor, which is excitatory, or it can be acetylcholine receptor. So that is something I want to highlight. So between this presynaptic cell and postsynaptic muscle, you have the structure that is present. It's called as a basal lamina, and that is playing an important role in synapse development. So we are going to talk about that soon. Okay, so basal lamina I talk about. So you have the receptor in the postsynapse called as acetylcholine receptor. This is a muscle fiber, and you have the junctional fold. So this is the real image. This is the EM image of neuromuscular junction, and this is the confocal image, the bright field image. This is the bright field image. This is the EM image. So here the green that you are seeing is the presynaptic active zone that I saw in the cartoon, like T-like structure. The green, these are the presynaptic cell, and the red is acetylcholine receptor labeled with alpha bungarotoxin. So this is a snake venom from the banded crate, which can target this acetylcholine receptor. So this preparation is incubated with the snake venom, the bungarotoxin. That's why it is targeting to the acetylcholine receptor. That's why you can label this acetylcholine receptor in the red. So that is what's shown in the confocal image. And here, this is the EM, again, the EM image of a neuromuscular junction showing the presynaptic cell. This is the postsynaptic cell. That is what is shown here, like how the neuromuscular junction look like. And the electron dense structure that you are seeing, the star, these are the active zone, okay, where the vesicle fuse and release its content to the postsynaptic cleft. So here again, comparing these two like kind of synapse. This is the neuromuscular junction between neuron and muscle, and this is the CNS between neuron to neuron. This is the central nervous system, and this is the neuromuscular junction. So here, this is the motor nerve, and this is the muscle. So the nerve sends its terminal, axon terminal, to the muscle, the innervates to the muscle, and this is what is shown. I've talked about that. So the receptor, the postsynapse, you have this receptor called as acetylcholine receptor, which is excitatory in nature, and presynaptic terminal, basically the terminal that you are seeing here, this is the axon terminal, and this is also surrounded by a Schwann cell which supports this axon like the presynaptic terminal. And this is the CNS synapse between neuron to neuron. This is a Purkinje cells. And then you have the dendrites, and then this is a cell body, this is the axon, and this is the cell body, these are the dendrites. And in the dendrites, you have the dendritic spine. You can see that in the dendrite, you have the spine-like structure. These are called a dendritic spine. And you have these structures is called as, this is the neck of the spine, and this is the head of the spine, which is also shown here. So this is the neck, this is the head, this is the basal, this is called a shaft. Okay, so this is the snapshot showing the dendritic spine. So again, you have this this is a tripartite synapse having the astrocyte and the presynaptic terminal and also the postsynaptic cells. So this is the postsynaptic cell, and this is the postsynaptic cleft, and this is the presynaptic terminal containing the active zone like the black or the dense electron dense structure having the mitochondria. So that is what is showing here, like the excitatory synapse are mostly acetylcholine containing receptor or glutamate receptor. The inhibitory synapse is the GABAergic receptor, the GABA containing neurotransmitter. Okay, so so this is again the typical structure, the mouse neuromuscular junction, and this is labeled with different marker that label pre and post synapse. So the postsynaptic membrane is red, and the presynaptic terminal is green, and yellow is the apposition between the they are opposed each other. The presynaptic and postsynaptic structure, they are opposed each other. That's why you see the overlap, the yellow kind of structure. Okay, so this is again shown here. The same thing, the presynapse, the green, and the postsynapse, the red. This is another another field. This is one field. So this is a mouse neuromuscular junction. So this is the mouse. Sorry, this is the mouse neuromuscular junction, and this is the CNS synapse. So I'm sorry for that. So this is the CNS synapse between the neuron to neuron, and this is between the neuron to muscle. So you can see that they oppose each other, pre and post synaptic compartment. They oppose each other. So So synapse, how the synapse is formed. So the next question is how they make synapse. They form the synapse with pre and post, how they come together, and they form the synapse. So I talked about the growth cone formation. You have this, presynaptic terminal, like from the growth cone, and the growth cone approach like grows and approaches to the postsynaptic cells, the postsynaptic compartment. So you can see that this is the growth cone, presynapse growing, and it is approaching to the postsynaptic cell or postsynaptic nerve. Then after that, you have generation of this basal lamina, which is shown here, and you have clustering of the receptor that is present in the postsynapse. These are the postsynaptic receptor. You can see that there is the clustering of receptor, and you have formation of this fold, the muscle fold, and you have even clustering of more receptor. Now you have formation of this dense-like structure in the presynapse, and you can see that the presynaptic active zone is opposed to the postsynaptic receptor. So they are formed in such a way that the presynaptic active zone, the electron dense structure, they remain opposed to the postsynaptic receptor, so that when you release vesicles, like synaptic vesicle, containing the neurotransmitter, releasing the presynaptic active zone, and it releasing the synaptic cleft and binds to this postsynaptic receptor to initiate the response, the signaling cascade. So the synapse formation is a bidirectional flow of information. So presynapse talk to the postsynapse simultaneously, postsynapse talk to the presynapse, so that you have a functional connection. Okay, so we are going to talk about how postsynapse talk to the presynapse to cluster certain molecules required for the synapse formation. Okay, so here showing that the same thing, like showing axon and the base formation of the synapse. So this green-like structure that you are seeing is the axon, and you can see that the receptor, the acetylcholine receptor, are marked with the bungarotoxin. This is the snake venom, so it targets the acetylcholine receptor. So I might ask question like bungarotoxin target which receptor, like acetylcholine receptor, glutamate receptor, GABA receptor, something like that. So it's just remember that the bungarotoxin basically target acetylcholine receptor to make it dysfunctional. So that's why you have labeling of this acetylcholine receptor. So you can see that the presynapse is expressing a fluorescent protein, so that you can see the green, and the postsynapse are labeled with bungarotoxin. So these are the yellow thing that you are seeing is the opposed person between the pre and postsynaptic compartment. Okay, so that is what, and here showing that the neuromuscular junction, connection between neuron to muscle. So one mature muscle cell have one connection from the forms one synapse. So you can see that here this is the motor neuron that are coming out from the from the central synapse, and these are the muscle fiber. So you can, this is one muscle fiber, another muscle fiber, and another muscle fiber. So it forms connection with each of the muscle fiber, and you can see that one mature muscle fiber, when it is in the mature stage, it forms one synapse. So here you can see in this muscle fiber, you can see that it forms one synapse when it is mature. So this is how you have formation of neuromuscular junction or connection between neuron to muscle. So one mature muscle fiber forms one connection. So that's what is showing here. So you can see that this is one mature muscle fiber. Here is forming two connection. You can see, but these are the terminally differentiated muscle fiber. So it's forming one muscle, one connection. Now we will talk about the postsynaptic differentiation, how the receptor in the postsynapse, they cluster together, forming a functional connection. Okay, so we are talking about the postsynaptic differentiation at the NMJ, and what the nerve tells the muscle. So here initially, when you don't have presynapse approaching to the postsynaptic cell, so you have a receptor present in the postsynaptic compartment. So before the axon innervates to the axon terminal, the synaptic terminal innervates to the muscle or postsynaptic cell, you have the receptor arises in the postsynaptic cell. So these are the receptor. You can see that these are the acetylcholine receptor, and you can see that you have around 1,000 receptor present in per micrometer square in the postsynaptic cells or postsynaptic compartment. But when the postsynapse see a presynaptic cell, you can see that they are uniformly, like they are distributed very scatteredly throughout this cell. But when it see the presynaptic compartment or presynaptic cell, they cluster together. Now you can see that there is 10,000 receptor cluster per micrometer square. So here postsynapse make adjustment according to the presynaptic cells. So here you have clustering of receptor when it saw this presynaptic cell. Okay. So you have clustering of receptor in the postsynapse in the past step. Then those receptors that you are seeing here, which are scattered in the in the postsynaptic cell, the half-life period is around 1 day. They are not stable, which are scatteredly distributed among these postsynaptic cell. But when you see the clustering of receptor, and the half-life became more stable. You can see that is the half-life period is around 14 days, because when it see the presynaptic cells. And how these receptors are formed. So you have because there is formation of a receptor, also there is a loss of receptor, because the half-life period is 1 day, here is 14 days. But how these are formed? So these are formed by local transcription and local synthesis of this receptor. So the local synthesis of the receptor happens when you have this kind of situation. So you have presynaptic cell, and when it's approaching to the postsynaptic cell, you have a clustering of the receptor, and the receptor only synthesized in the the nucleus, which is present below this junction. So you can see that the synaptic transcription and translation happens during in this junction. The nucleus, the muscle cell is a multinucleate cells, so there are many nucleus in the muscle. So the nucleus which are present down to this or below this to this connection, they are actively transcribed to get this receptor. But some of these nucleus which are present in this region, they are not that active. They also can synthesize the receptor, but very less concentration. So these are called as extrasynaptic postsynaptic receptor, the acetylcholine receptors. Mostly the nucleus which is present here, they are actively transcribed, because they get signal to transcribe and to produce more receptor, because you need the clustering of receptor in the postsynaptic compartment. Okay, so that is what how you have clustering of a receptor and formation of postsynaptic receptor in the muscles. So, and also it has been shown that nerve activity can also regulate expression of acetylcholine, the postsynaptic acetylcholine receptor in the neuron. So this is what is shown here. So when this is a neuron, and here this is the presynaptic terminal, the presynaptic nerve, and this is the postsynaptic cell. So you have this clustering of this acetylcholine receptor. These nucleus are very active, and they produce the postsynaptic receptor. These are actively repressed. They are not active. These nucleus that you are seeing here, they are not that active to produce this receptor, because you need because these get signals so that they can produce extra acetylcholine receptor. So these nucleus are actively they are not active. They are repressed. But when there are two situations, you have denervation, you have renervation. Denervation means you have loss of the nerve, that is called a denervation, and reinnervation means you have you have formation of synapse, so that is called innervation. So in this situation, when there is denervation, when there is loss of this nerve connection to the postsynaptic cell, these receptor, this nucleus which are present in the in this region, they are actively they become active, and they actively transcribed, and you can see that they become active. So here it is saying that nerve activity represses extrasynaptic receptor. So when you stimulate this nerve which is denervated, so this is denervated and this become active. When you stimulate this nerve, or you stimulate this preparation, you can see that you see the active, you can see that they become repressed. So that's why the nerve activity actually represses the transcription of acetylcholine receptor. That's what I want to say. Okay, so here you can see that. So when it is denervated, when you have loss of the nerve connections, so these become active. Those nucleus which are present in this side, not in this side, become active. Those are actively transcribed acetylcholine receptor. Now, when you stimulate this preparation, they become repressed. So that means when there is, when you increases the nerve activity, they become repressed. That's why it's called as nerve activity represses extrasynaptic acetylcholine receptor. Okay, because you have only accumulation of the acetylcholine receptor which have a functional connection. The other side you don't have this functional connection. That's why those receptor they become inactive. You do not need the receptor here to be inserted. Okay, so only you have insertion of a receptor in this region. Okay, so that's what kind of point say that when you stimulate the nerve, the extrasynaptic the nucleus which are present in the extrasynaptic region. So this is the extrasynaptic region, this is the synaptic region. The extrasynaptic region, the nucleus which are present in the extrasynaptic region, they remain repressed. They do not produce more acetylcholine receptor. However, the nucleus which are present in the junction region, they become active. They produce more acetylcholine receptor to form a functional connection. That's what I want to highlight. So then the next question is how these acetylcholine receptor are clustered. So we are saying that when you get this nerve, you have clustering of the acetylcholine receptor in the postsynaptic cells. You get around 10,000 receptor per micrometer square. But how this thing is happening? What is the model, or what is the signal? So this has been studied in torpedo electric organ. So this is evolutionary derived from muscles. So the torpedo electric organ is evolutionary derived from the muscle and is extremely rich with acetylcholine receptor. So this is the torpedo Californica fish. So this is and you have this electric organ which is enriched with acetylcholine receptor. So this electric organ is the evolutionary derived from the muscles. That is want to say. So this looks like this. So and in this synapse, it has been shown that there is a chemical which actually induces the accumulation of postsynaptic acetylcholine receptor. Those chemical is the agrin. So agrin stimulate the initially people have thought that now the model is changed. So I will be talking about that. So the agrin actually helps in clustering of this acetylcholine receptor in the postsynaptic cells. So here they have done the experiment. This is the control, and here what they have done is they have taken the basal lamina of the NNJ preparation, and they have incubated the torpedo electric organ extract to this NMJ, the neuromuscular junction. And you can see that in the control, when they don't incubate, they do not see this acetylcholine receptor, accumulation of acetylcholine receptor or clustering of acetylcholine receptor in the NMJ, neuromuscular junction. However, when they incubate torpedo electric organ extract, you can see that you have accumulation of this acetylcholine receptor. That means this agrin, this torpedo electric organ extract contain these chemicals, the agrin chemical, which induces clustering of this acetylcholine receptor. Okay, so that is one to say. So it is a very complex signaling pathway where agrin actually signal through this receptor complex called as MuSK LRP4 receptor complex, and that complex actually helping in clustering of the acetylcholine receptor. So this is the membrane, and here is the receptor, and this is the ligand, agrin ligand. So the agrin ligand bind to the MuSK receptor having the LRP4, and now you have activation of a downstream signal and the activation of the signaling cascade that is required for the actin regulation, actin dynamics, and
through this signaling pathway, through the actin dynamics pathway, you have clustering of the, you have trafficking of the acetylcholine receptor. You have insertion of acetylcholine receptor to the postsynaptic compartment. Also, you have acetylcholine receptor immobilization. Okay, that is showing here how the agrin MuSK LRP4 pathway through regulating the actin cytoskeleton, helping insertion of the acetylcholine receptor or clustering of the acetylcholine receptor to the postsynaptic compartment. So this is what is shown here. So this is the presynapse and this is the postsynapse, and you have this receptor, the acetylcholine receptor, in the postsynaptic compartment. Now the presynaptic cell secretes this chemical. Mostly, it is believed that the presynaptic neuron secretes this chemical called agrin, and because of secretion of agrin, you have the initiation of this downstream signaling, the MuSK LRP4 activation, and which induces actin pathway, and it requires for the clustering of this receptor. So you can see that the MuSK LRP4 receptor actually, through this pathway, it regulates another molecule called the rhabsin molecule. Okay, through this actin dynamics pathway, it regulates, it stimulates a rhabsin molecule to be recruited, and this rhabsin actually helping in clustering of the acetylcholine receptor to the postsynaptic cell. That is what is shown. Now, here some studies are done by knocking out or knocking down different components of this pathway to see whether you have formation of synapse or not. So this is a neuromuscular junction. This is a wild type synapse. So you can see that these are the axon, these are the receptor in the red, the postsynaptic acetylcholine receptor. You have normal acetylcholine receptor in the wild type. But when you knock out or when you remove the agrin, in the mouse, or you delete the agrin in the mouse, the knockout mouse, where you don't have the ligand. That is, agrin is a ligand. It binds to the receptor, MuSK LRP4 receptor. You can see that you don't have this, functional, formation of this receptor or connection, or you don't have this proper synapse formation in agrin knockout, or in the receptor knockout or MuSK or LRP4 knockout. You don't have this, functional connection. Or when you knock out the rhabsin, basically rhabsin recruits the, acetylcholine receptor. In this situation also, you do not see formation of the functional synapse. So knockout of agrin or its receptor or rhabsin perturb NMJ formation of synapse. Okay, so now this is what is believed in this area, but they always, there is some exception. So you do not see, you cannot generalize that agrin or rhabsin always do this to cluster acetylcholine receptor in the postsynaptic cells. There are always exceptions to the hypothesis. So here there are some hypotheses which suggest that agrin is not always required for clustering of acetylcholine receptor. And you know that agrin secretes from the presynaptic terminal. So here it has been shown that in the zebrafish model, this clustering of the receptor happens before the formation of synapse, the formation of functional connection. So here you can see that this is the presynaptic cell, which is in the green, and in the postsynaptic muscle, you do not see the axon, right? The green things here, but you still have the formation of the postsynaptic acetylcholine receptor. So you do not have the presynaptic cell. That means you do not have the agrin. Then how this is happening? So this is an exception to this hypothesis. We are going to, there is a model explaining how this is happening. Even you don't have this agrin, you still have formation of, acetylcholine receptor. Okay, so that is what's in the zebrafish model. So this is a time-lapse image showing that, even the acetylcholine receptor cluster from the, you have the generation of the acetylcholine receptor prior to the arrival of the nerve terminal to the muscles. That means you have this receptor clustering before the secretion of agrin chemicals from the presynaptic terminal. So, okay. So here another example from the mouse model. So here this is a wild type synapse. This is the green thing that you are seeing is this is the wild type mouse neuromuscular junction. The green that you are seeing is the motor neuron axon terminal. and the red that is seeing here in other channel is the acetylcholine receptor, and here is the knockout mouse, knockout for the presynaptic terminal. So they knock out this transcription factor HB9, which is a motor neuron transcription factor. So when they knock out this HB9, you do not see the presynaptic compartment or presynaptic motor neuron. So you have loss of presynaptic motor neuron, but you can see you still have the postsynaptic acetylcholine receptor. You don't have agrine here. You don't have the motor neuron, but still you have the formation of this, acetylcholine receptor. So how this is against the hypothesis that we saw, just a few slides back. Okay, so there are some hypotheses. So we'll talk about this thing, like how we'll talk about this, how this thing happening. You don't have this agrine, how this is, you have the clustering of the acetylcholine receptor. So before going detail to that, just want to show some of these things, how acetylcholine is synthesized and packaged in the vesicles. So this is it forms the chemical reaction between the acetyl coenzyme and the choline, and you have this enzyme called as choline acetyl transferase. This is called a CHAT, choline acetyl transferase. So when you have defect of this enzyme, choline acetyl transferase, you do not have formation of acetylcholine. And when you have this enzyme, you form this acetylcholine. So after formation of acetylcholine, they are transported, they are packaged in the vesicles, and you need the transporter. So this is called as vesicular acetylcholine transporter. So you need the V-CHAT to transport this acetylcholine to the vesicles, and they remain packaged. So they fused to the presynaptic terminal, that is the active zone, to release its content. So this is just want to show that how acetylcholine is synthesized and packaged in the vesicles. Okay, so here again we'll come to this agrine hypothesis. The agrine hypothesis says that when the presynaptic neuron approaches to the postsynaptic cell, it secretes the agrine, and because of secretion of agrine, you have activation of this pathway, MuSK LRP4 pathway. Now that helps in clustering of this acetylcholine receptor. That is the agrine hypothesis. Now we have also shown that when you don't have presynaptic neuron, or you still see the receptor clustering. Now this is the against agrine hypothesis. So this needs to be modified. Okay, so we are going to talk about that in the latter side. But here this is just showing that without agrine, you can see the clustering of postsynaptic receptor. Okay, so here there are 4 genotypes shown. One is the control, so the presynaptic neuron, the green, the postsynaptic receptor, the acetylcholine receptor, in the red. So the agrine minus, that means you don't have agrine. So you can see that you still have acetylcholine receptor. So here you can see that you have less acetylcholine receptor cluster, which is shown compared to that control. You can see that there is a reduction of acetylcholine receptor. So this is that is what is in the previous slide I have shown, that agrine required for the acetylcholine clustering of acetylcholine receptor. Now here you can see the knockout chart. That is choline, what is CHAT? That in the previous slide I talked about CHAT, the choline acetyl transferase, this enzyme. When you don't have this enzyme, you don't have formation of acetylcholine. Now you don't have formation of acetylcholine, so acetylcholine will not be released. The acetylcholine will not be released means you do not have activation of postsynaptic receptor. So the postsynaptic receptor actually, cluster depending upon the signal from the acetylcholine. Okay, so you need the acetylcholine to so that you have more clustering of the postsynaptic receptor. So when they knock out the CHAT, that means you don't have a synthesis. Sorry. Yeah. So yeah, that's what I want to say in the slide. So as I mentioned, there is always exception. So in some cases, in the zebrafish model, it has been shown that agrine is not required for clustering of the acetylcholine receptor. But in earlier slide, I talked about in the mouse and MJ, you require agrine first in the clustering of the receptor. So now what they are doing, they're manipulating different components. They're manipulating acetylcholine, they're manipulating the enzyme, they're manipulating the transporter, and want to see how this clustering thing is happening, what is the pathway they want to dissect. Okay. So here they are knocking down this enzyme or knock out the enzyme, the chart, which is required for the choline acetyl transferase, required for the formation of acetylcholine. So here, agrine minus, you see reduction of the acetylcholine receptor. Also, you see the reduction in the innervation acetylcholine receptor, and the cluster size is also reduced. And the chart knockout, where you delete the enzyme, you do not have formation of acetylcholine, you still see the clustering of the acetylcholine receptor here. Okay, you can see that, it's like even more than the control. But when the double knockout chart and agrine, you can still see the formation of this, acetylcholine receptor. So how this is happening? Blocking acetylcholine release or knocking out acetylcholine transferase, rescue this agrine knockout phenotypes. You can see that. So this is a double knockout. Double knockout means they knock out agrine and then they knock out chart. So that is the double knockout. So we know that agrine minus has reduced acetylcholine receptor, which is shown here. Now the chart minus, you remove this enzyme, you still see the acetylcholine receptor clustering. So here no receptor clustering, here receptor clustering. When the double knockout, they see the clustering of receptor. That means the chart enzyme or the chart minus is rescuing this phenotype, agrine phenotype. That's what's shown here. Okay, so they're just dissecting the pathway. In the coming slide, I will show you that how this thing happening, what is the mechanism for this clustering of this receptor. So here they eventually found that the chemical, the molecule, the DOX7 molecule, actually required for the clustering of the receptor. So I will show you in the coming slide what is the role of agrine, what is the role of DOX7, how it is helping clustering of the receptor. So here this slide shows that the cytoplasmic protein DOX7 is a cytoplasmic muscle protein. It binds to the MuSK, the receptor for the agrine, okay, MuSK, and activate it and induces acetylcholine receptor clustering. So here is the kind of experiment they have done. So here they have taken these muscle cells, okay, and they incubate this DOX7 molecules, and they saw that you have clustering of the acetylcholine receptor in the muscles. Okay, so they say that, and also the DOX7 is colocalized with acetylcholine receptor. So the DOX7 actually induces acetylcholine receptor clustering at the post-synaptic compartment. Okay, and then what is the role of agrine? So we will going to talk about that in the model. Okay, so here again they have done the experiment with the DOX7. So they have done the experiment at different time point at E14.5 or E18.5. So this is the wild type synapse. This is the knock, and this is, you can see that you have acetylcholine receptor clustering. And when the knock out the DOX7, you do not see the clustering. But in other synapse, I show you that when the knockout agrine, sometimes in some cases you see the clustering of receptor, in some cases you do not see the clustering of receptor. But however, the DOX7 is in every cases you do not see the clustering of receptor, when you knock out DOX7. So you can see that you don't see the clustering of receptor at E14.5, or you knock down or knock out at the latter stage at E18.5. You can see that you do not see the clustering of acetylcholine receptor. That means in DOX7 is required for the clustering of acetylcholine receptor. Okay, so it is, and just recall this agrine hypothesis. So we saw that agrine is sometimes is required for the clustering of receptor, and sometimes in some NMJ is not required. But DOX7 is it's consistent in all the cases. It is required for the clustering of acetylcholine receptor. Okay, so DOX7 mutation inhibits clustering of acetylcholine receptor either at the earlier stages or at E14.5 or E18.5 in the latter stages all the time. Okay, so this is again kind of showing the same thing, that the rescue of this agrine phenotype with double knockout, the chart choline acetyl transferase. That is what is highlighted here, double knockout. Okay, so here is the model. How the current we just modified this agrine hypothesis. The current explanation for NMJ formation is you required both of these agrine and most importantly DOX7 for clustering of acetylcholine receptor. So this is the A model. You can see that you require the DOX7. So DOX7 activate the MuSK receptor, and the MuSK receptor, again activate the rhabsin required for the acetylcholine receptor clustering. So this is what is shown here. But what happens when this happens before innervation, prior to the innervation? So you do not see the neuron, and in the muscle this is what is happening. In the muscle you have formation of DOX7, then this activate the MuSK receptor. Now the MuSK receptor recruits rhabsin or activate rhabsin, that activate the pathway which is required for the acetyl receptor clustering, and that happens prior to the innervation, prior to the formation of the synapse. Now when the postsynaptic cell see the neuron, the postsynaptic presynaptic neuron, so you can see that now you have this agrine which is released from the motor neuron. Now this again binds to the MuSK in combination with the DOX7. It activates the rhabsin, and rhabsin actually helps in clustering of this acetylcholine receptor. Also this motor neuron secretes acetylcholine because it has this it has this like presynapse you have this acetylcholine, so it secretes acetylcholines. And acetylcholine, what it does actually, acetylcholine it forms this receptor, the acetylcholine receptor. That's what I said, that acetylcholine can also induce clustering of the acetylcholine receptor in the postsynaptic compartment. So when the presynaptic neuron secretes these acetylcholines, so acetylcholine diffuses to the postsynaptic, it releases in the synaptic cleft, and then it diffuses to the postsynaptic compartment. It can also induce the clustering of this receptor. Now this clustering, this is called as extrasynaptic clustering, because you don't have a synapse here, you have a synapse here, neuron and muscle. Now this is inhibited by this agrine. So agrine, what agrine does is, agrine restrict this extrasynaptic clustering. It inhibits extrasynaptic clustering. So the DOX7 actually required for the synaptic clustering, and agrine, what it does is inhibits the extrasynaptic clustering. So that is what the current hypothesis is. You have formation of pre and postsynaptic like connection between the pre and postsynaptic cells and clustering of the receptor. So this is what is the current model. The current model is this MuSK DOX7. They regulate the clustering of acetylcholine receptor, and when you have secretion of acetylcholine or in the presynaptic compartment, they can also randomly can also insert the receptor in the postsynaptic compartment or postsynaptic cell, and this is being inhibited by the activation of agrine. So agrine actually stops this. Okay, so that is the current model. So it is showing that the same thing, showing that positive signal, like positive pathway through this agrine, not agrine, so positive pathway through this MuSK and DOX7 required for the clustering of this receptor. And when you have activation of the neuron, what happens is electrical activation, means you have secretion of this acetylcholine, release of acetylcholine, so not secretion, the release of acetylcholine, that happens because of electrical stimulus. And when you give electrical stimulus, you release acetylcholine, and acetylcholine can randomly insert acetylcholine receptor in this compartment in the postsynaptic cell. Now agrine become activate and stop these processes. Okay, so that is what is shown in the model, showing that how you have formation of these receptor clusters. Okay, so here it is showing that postsynaptic differentiation at the NMJ. Like we talk about more about the presynaptic compartment. Now we are going to talk about how the postsynaptic cell talk to the presynaptic cell. So here this is showing that different scenario. So here this is the muscle and this is the neuron. So you can see that is the formation of neuromuscular junction. And here you have several cases. You can also kill these muscle cells, or also you can denerve the muscle, the neuron. You can also kill the neuron. But even if you kill the muscle cells or you kill the nerve which is innervating to the muscle, you still can see the formation of functional connection. So this is called as a regeneration. Okay, this type of degeneration that you are seeing is called as Wallerian degeneration, and this type of regeneration, even after killing this neuron, like the you can cut this, cut this axon or kill these muscle cells, it can still form the contact. This can also regenerate. Okay, so that means the postsynaptic cells actually sending some signal to the presynaptic neuron that can actually again make the connection. Okay, so that is called as regeneration. And the signal is coming from this muscle that is called that is present in the extracellular matrix. So we are going to talk about what are the signals I know that actually talking to the presynaptic cells or presynaptic neuron, so that presynaptic neuron again can grow and form the connection. Okay, so we'll talk about some of these component in the extracellular matrix, which is the laminin protein that form the basal lamina. In the very beginning of this my lecture, I talk about today lecture the lamina, basal lamina. So it is a trimeric, heterotrimeric protein, consists of 3 different chains like alpha chain, gamma chain, and beta chain. So you can see that you have 3 different type of chains. So it's a trimer. So consists of 5 different types of alpha chain, 4 different type of beta chain, and 3 different type of gamma chains. So you can make a lot of combination. You can have alpha 5, beta 4, gamma 1, something like that. You can make a lot of combinations. So there are 60 combinations you can make, and in the in vivo model it has been shown that there are 15 different trimers which are present, but you can make 60 different combinations. Okay, so that is shown here, the laminin protein, the present in the extracellular matrix, the muscle cells, which talks to the presynaptic cells to form this connection, the functional connection. So here showing that laminin. So this is the presynaptic nerve, and this is the more postsynaptic muscle fiber, and the postsynaptic, the neuromuscular junction, having this predominantly this type of laminin present in the neuromuscular junction. You have alpha 5, beta 2, gamma 1. So that is called as beta 2 containing laminin, that is called as S-laminin, is present in the neuromuscular junction. So you can see that the laminin are present in the postsynaptic cell, and that actually gives signal to the presynaptic cell to make a functional connection. Okay, here some of these studies, the alpha 4 knockout mice or knockout cell, you can see that you have loss of this active zone in some cases, and in the beta 2 knockout, beta 2 laminin knockout mouse, you can see that there are less of the folds. The folds that you are seeing here, these are called as muscle folds, are missing here. You have some defect when you have loss of this laminin, either alpha laminin, beta laminin, or gamma laminin, and there are different versions like alpha 1 to alpha 5, beta 1 to beta 4, or gamma 1 to gamma 3. Okay, so that is shown here again, the same thing. And laminin is a very important molecule and is required for the clustering of voltage-gated calcium channel. And voltage-gated calcium channel is present in the presynaptic cell, required for the influx of calcium for fusion of the synaptic vesicles. So here this is the active zone. Okay, active zone means where the vesicle fuse, and it consists of multi-protein. So you have, these are the presynaptic protein. In the Drosophila it is called as Bruce pilot. In the mammal it is called as cast ELKS. You have ring binding protein, you have piccolo protein, you have boson protein. They all make the active zone component. Active zone is a multi-protein component. Okay, it's an electron dense structure where the synaptic vesicle fuse and release its content. And also you have in the voltage gated calcium channel present close to this active zone, which is required for the vesicle fusion, because when you give the stimulate the nerve, you have influx of calcium, and this calcium actually influx through this voltage gated calcium channel, and this voltage gated calcium channel is recruited, or is organized by this laminin that I talked about, present in the muscle. Okay, this laminin, alpha 5, beta 2, and gamma laminin present in the neuromuscular junction, the S-laminin that is shown here. So that is what. So these are the key points. Acetylcholine generates electrical activity, or action potential. Agrin is required for the proper clustering of acetylcholine receptor, and you require this S-laminin for the organization of the voltage-gated calcium channel in the presynaptic compartment. So we'll quickly go to the synaptogenesis in CNS, the central nervous system. So these are some key difference. So this is the NMJ, neuromuscular junction. You have this presynaptic cell in the postsynaptic cell, the muscle, it forms the fold, and you have this laminin protein, which is required for the clustering of or organization of the calcium channel. Now you have this active zone. In this CNS, synapse, where you have between the neuron to neuron, the difference between the neuron to neuron synapse or neuron to muscle synapse is CNS synapse. The CNS synaptic cleft has no basal lamina. So here you can see that NMJ has the basal lamina. You do not have the basal lamina in the CNS synapse. Other thing is that agrin is not critical in the CNS synapse. Agrin is critical in the muscle neuromuscular junction, but not in this synapse. Okay, and the CNS synapse also use different neurotransmitters. So you have GABA type neurotransmitter present in the CNS synapse, and different molecular components are also present in the CNS synapse, which are not present in the neuromuscular junction. So here showing that CNS synapse. Okay, so this is a excitatory synapse, and this is a this is inhibitory synapse in the CNS synapse. So glycine receptor is an inhibitory synapse. So here, for clustering of glycine receptor in CNS synapse, you require Zephyrin. But for clustering of acetylcholine receptor in the muscle, you require DOX7, you require agrin in the NMJ synapse. But in CNS synapse, you require Zephyrin for inhibitory synapse clustering, the glycine receptor. And for the clustering of ionotropic glutamate receptor, the excitatory synapse in CNS, you require PSD95, okay, or PDZ containing protein. So I'm going to talk about what is PDZ containing protein. So PSD95, so this is again the neuromuscular junction. Sorry, I talked about wrong. This is a neuromuscular junction. The inhibitory synapse. This is not inhibitory. Sorry, I'm so sorry for that. I confused with the glycine receptor. So this is the acetylcholine receptor. I'm just comparing with two different things: excitatory synapse and neuromuscular junction. So in the excitatory synapse, the acetylcholine receptor in the neuromuscular junction, you required rhapsine or MOSC receptor or rhapsine or agrin. Okay, but for clustering of CNS, ionotropic glutamate receptor in the CNS, you require the PSD95 or PDZ containing protein. So this is the excitatory synapse. This is also excitatory synapse. This is excitatory synapse in the NMJ. This is excitatory synapse in the CNS. In the CNS, you require PSD95, PDZ. In the NMJ, you require rhapsine. Okay. So again, I'm showing another type of inhibitory synapse I was talking about in the previous slide. This is inhibitory synapse. Here, this is a GABAergic inhibitory synapse or glycine inhibitory synapse. So you require Zephyrin for clustering of this glycine receptor or GABAergic receptor in the CNS. Okay, or in the excitatory synapse in the CNS, you require PSD95 or PDZ. So here, some of this protein, PDZ containing protein required for the clustering of this, clustering of this NMDA type glutamate receptor in the CNS. So now we are in the CNS. Okay, so these are a variety of protein. So this has a PDZ domain, which PDZ means PSD95. P stands for PSD95, and D stands for disc large, and Z stands for zonula occludens. So they have the PDZ containing protein. They have this domain, conserved domain. That's why they are called as PDZ containing protein. Those PDZ containing protein are listed here. You don't have to remember these things. And this is required for the clustering of NMDA type glutamate receptor in the CNS synapse. Okay. So here this is the NMDA type receptor, glutamate receptor. So you require this PDZ containing protein. And also PSD95 or PDZ containing protein for clustering of this glutamate NMDA type glutamate receptor. Okay, that is what is shown here. And also other molecules like neuroligin molecules, they also require this PSD95 or PDZ containing protein for the clustering of this other cell adhesion molecule, the neuroligin molecules. Okay, so the PSD95 or PDZ containing protein, they are a scaffolding protein. They require they helps in assisting this receptor or this the cell adhesion molecule to properly function in the compartment. The postsynaptic compartment is showing that how the Zephyrin is required for the clustering of glycine receptor, the inhibitory synapse, or the GABAergic receptor in the central synapse, the CNS synapse. Okay, So Zephyrin is not a PDZ containing protein, but it can also induce the clustering of the glycine receptor or GABAergic receptor. So here this is showing that how Zephyrin knockout the Zephyrin knockout mouse can require for clustering of glycine receptor. So this is a wild type mouse having glycine receptor and glutamate receptor intact. You can see that this is the diagrammatic representation showing that the glutamate receptor or glycine receptor in the CNS synapse. And when you knock out Zephyrin, which is required for the glycine receptor clustering, you can see that in the knockout mice, you do not see clustering of glycine receptor. You do see clustering of glutamate receptor. So glutamate receptor is there, the glycine receptor is missing. That means Zephyrin is required for the clustering of glycine receptor in the CNS synapse. So this is some of the image showing that how you can see the abnormalities in the mice. This is the Zephyrin wild type mouse. So you have one copy of Zephyrin, seems normal. You loss of the Zephyrin, you can see that they are abnormal, and these phenocopies with this styricine kind of like phenotype. So in the styricine mouse, you can see that you have they are abnormal compared to the wild type. So they resemble like the styricine mice. So the Zephyrin knockout mice resemble like styricine mice. So they have a lot of abnormalities because you do not have clustering of glycine receptor. Okay. So this is I have only one or two slides. So this is showing that 80% of excitatory synapse, which is the acetylcholine type, are on the dendritic spine. So here, this is the cell body, and this is the axon, and this is the this is the shaft for the axon, the dendrites, and this act as a shaft, and these are the shaft, and you have the spine coming out from this dendrite. It's called the dendritic spine. You have this kind of acetylcholine kind of receptor present, or in the CNS, you have this NMDA type glutamate receptor are clustered in this region. So acetylcholine type receptor present in the neuromuscular junction, and you have NMDA type or AMPA type glutamate receptor present in the CNS synapses. So mostly in the dendritic spine. So this is the shaft, dendritic shaft, the base of the dendrite. This is the shaft of the dendrite. So you can see that this is the neck. This process is called neck, and this is the head of the dendrite. And you have the presynaptic terminal, the axon terminal, that is making connection with this dendritic spine, and this is the active zone where you have this vesicle fused to this active zone to release its content in the postsynaptic compartment. Now in the postsynaptic compartment, you have the receptor, either glycine receptor or NMDA receptor or AMPA type receptor. Now after binding to this neurotransmitter, they become active and initiates the downstream signaling process. Okay, so this is again the image showing that different compartment is a dendritic spine. So this is a presynapse, axon terminal. This is the postsynapse. You have postsynaptic density. Those proteins like PHD95 or PDZ protein, they forms the postsynaptic density. And this is the postsynaptic cell. This is the endoplasmic reticulum, sarcoendoplasmic reticulum. And this is the glial cells, the astrocyte type glial cells. This is a tripartite synapse. Okay. So here showing that how dendrites are formed. So this is and how this forms a connection to the another neuron. So here this is the axon. So this is the axon terminal, and this is the cell body. Here cell body will be here. Now from the cell body you will have dendrites, and from the dendrites you have dendritic spine, and dendritic spine making connection to the axon terminal. That is what is showing here. So those dendritic spine, they are formed from this dendrites. They form in three different structures. So here they form as like a protrusion, phylopodium. So here these are the phylopodium-like structure. They are very transient. So they don't have. You can see that you have the cell membrane which is green, and you do not have. This is a phylopodium. This is a protrusion kind of structure forms from the dendrite. You have this green, the cell membrane, the presynapse, but you do not have postsynaptic component, the PHD95 or all these things. So you do not have these things, postsynaptic densities, the PHD95 or PDZ protein or the receptor. So philopodia do not have these things. So you have formation of when you have formation of dendritic spine, first it extends its process, forms philopodia. Now it forms protospine, which have postsynaptic receptor. Now the protospine becomes stable to form the stable spine, and the stable spine contains the receptor, postsynaptic receptor, and presynaptic compartment, the presynaptic cells. that's what kind of generation of dendritic spine from the dendrites. So this is the last slide. This is showing that formation of protrusion, the philopodia. So you have the presynaptic part, you do not have the postsynaptic part, the PSD95 or the receptor. But when it forms the mature spine through protospine, it forms the protospine. So you have formation of the clustering of the receptor in the protospine stage. Now, when it becomes mature spine, you have formation of functional receptor in the postsynapse. Now, in the presynapse, you have the axon, you have the terminal, you have this formation of the active zone. That is how you have the formation of synapse. Okay, so that is all about synaptogenesis. So let me know if you have any questions. So we'll move forward with that. Thank you very much.